right, here we go. Question 4. Question 4 is one of those where the actual wording of the questions might have confused you somewhat. So we will look at I will look at reading and interpreting the question much more than actually looking at the individual questions later on. So you have got somewhere in that you're somewhere in the metabolic pathway that happens to produce ATP in respiration. Part of this is glycolysis, part of this would later on link to link reaction and what happens in mitochondria later on with the Krebs cycle and the electron transport chain. But we're here in the initial stages. We're here in glycolysis. So it doesn't actually give you the name of hexokinase and phosphofructokinase, it just shortens it down for you. So fancy enzyme here that helps phosphorylate glucose a first time round, then the second enzyme helps with the phosphorylation. A second time round, hexose bisphosphate. So you've got 2 phosphates on it, in which case now it splits into your 2 trioses, your pyruvate that is later on needed for follow-on metabolic pathways. It also gives you if we continue going down this route, this is the box standard aerobic respiration, as in we're using up the glucose within the cells to release energy, to release those ATP. That would be the pathway downwards. This other pathway here, think of this as what would be happening in the liver. It also happens in muscle cells and happens in all other cells as well. You've got a storage compound, glycogen. You know this compound from your biochemistry. So you've got an alternative pathway that would lead to the storage of glucose. You will know from whatever you know about diabetes, if there's too much glucose in your bloodstream and it can't get stored away, that excess glucose will just leave the body through urine because you don't have enough absorption capacity in your proximal convoluted tube, I believe it was, but you'll do kidneys later on. Okay, it then gives you lots of information. The control of glycolysis control means how fast each process is allowed to take place. If there are high levels of ATP, that means there's plenty of ATP in the cell already, and you don't need to make even more ATP. You cannot store ATP. So it would be making more sense to store the excess glucose in a different version. So if there is plenty of ATP, then this enzyme here, the second enzyme, the phosphofructokinase, that's the one that is being inhibited. It's inhibited allosterically, and therefore the shape is no longer right. The affinity for the substrate, which was the glucose 6-phosphate, no longer fits the active site. And it's the main regulatory enzyme, but it's not the only one. Hexokinase is also catalyzing the first step, and it is inhibited by its own product. So it's making glucose 6-phosphate, and then glucose 6-phosphate inhibits the actual enzyme. So it's kind of I've made something, but that something that I've made is still stuck in the active site and I can't let go of it just yet. So it's a follow on and follow on and follow on. So the first question should be straightforward. Allosteric inhibition. Please don't describe the word allosteric with allosteric. You will need to say that it's a place on the enzyme that is not the active site, on a different site of the enzyme. And you will need to say that the shape is changed. The active site can no longer fit onto the substrate, and therefore the enzyme is no longer functional. As mark scheme, it looks like that. Inhibitor binds to a site, active site, away from the active site. Okay, changing shape, substrate no longer fits. Then let's look at the next one here. Explain why the cell uses the inhibition of that second enzyme rather than the first one to slow glycolysis. Why do you not want to slow it down at the first enzyme? Why slow it down at the second enzyme? What can we do after the first enzyme then we still may want to do? Oh, we may want to still store the glycogen. We are slowing down the phosphofructokinase because then we can still use the glucose 6-phosphate to be stored as glycogen, and we're not just losing the glucose we had. So mark scheme to go with that. Hexokinase is that one here. Inhibiting PFK means that glucose can be converted to glycogen and it allows the storage. Okay, you can argue the same thing just the other way around. If you inhibit it the other ways means that you can't store, okay? So you can argue both ways around. It's many ways leading to Rome situation. Okay, moving on to the next one. Again, it's confusing you with some big fancy compound. 3-BRPA is a chemical which inhibits, in this case, HK hexokinase. So that very first step cannot take place. A small chemical group is added, you've got a change in shape, the enzyme no longer works. So now this enzyme has a potential use. So the chemical which attaches to that enzyme has a potential use as an anti-cancer cell. It seems to slow down the entire glucose breaking down pathway, so the glycolysis pathway, right at the very beginning. All cells rely on this pathway. All cells do. However, cancer cells more than others. They rely heavily on ATP from glycolysis. So it's literally a cell death. How does that work? If you're inhibiting this process, you're not going to get glucose 6-phosphate, which means you're not going to be able to continue the entire pathway. Let's have a look at the mark scheme together. So it's no longer able to bind with glucose because of that extra chemical that has been attached, and the active site has changed, which means glucose 6-phosphate can no longer be synthesized, and therefore no ATP is produced, which means that cells will die. Healthy cells as well. So I'm assuming if a patient was actually given that compound, that fatigue would be one of the side effects. I'd have no idea. I wouldn't need to look into the research for that one. Okay. A follow-on question here. It's actually a whole lot of preparation for that question, when all it wants is why is this ice cold isotonic buffer? What does that mean? 3 marks, ice cold isotonic buffer. The entire rest is a description of one of the practicals that you did with yeast and the methylene blue as a hydrogen acceptor in that one. Ice cold, what doesn't work well at cold temperatures? Oh, we've got enzymes that don't work good at cold temperatures, therefore might have an effect on cell membranes. Isotonic, you may not want different salt concentrations on opposite sides, different water potentials, and therefore parts of the cell that you actually want to investigate and look at. If you want to look at pyruvate digestion, you might want to look at mitochondria, so your mitochondria might be lysing. That's not a good idea. Buffer, that is a chemical to keep the pH steady, to keep the pH at whatever value you have it set. Usually it's bicarbonate that's being used. So have a look. Ice cold, enzyme activity slowed down, so disruption of membranes. Isotonic, water potential is the same inside and outside, so you've got no lysis happening. And the buffer is to stop pH changes. Okay, nearly there. Pyruvate. Why is it used rather than glucose? What process are we looking at? If we're looking at a process in the mitochondria, which may no longer be glycolysis, which may be link and then the Krebs cycle. Let's have a look. Isolating liver mitochondria. So what process is happening in mitochondria, which is then everything that follows on after glycolysis. So link reaction and Krebs cycle that happens in there. Pyruvate is the right fuel for that. Glucose isn't, quite simple. So mark scheme, because glycolysis takes place in cytoplasm, and pyruvate is the right substrate for mitochondria and the link reaction in there. Right, nearly there. I said that already, didn't I? Okay. Did we do this one? Yes, we did. Okay. This question, it gives you a set of results, the way you should be writing up your lab report. So you've got a temperature range for your experiment, the 3 trials, the mean of your 3 trials, and the standard deviation. Now, some of you might be rather confused what is meant by standard deviation. I never got you to calculate it, and I'm not quite sure if Katie did. Excel does it for you in one click. You just put in your numbers, stdef, a function stdef for standard deviation. What does it actually do? I've just, on this extra tab here, I've got the equation off for you. It looks a bit scary at the start, but actually all it would be, each value that you have, how far is it away from the mean? That is the gist of standard deviation. For each single value that you've got calculated, how far is it away from the mean value? So if you go back to your data set, each of those, if that's the mean, what's the difference between those two? So basically 332 take away 385, and then add that with 332 take away 290 plus 332 take away 320. Do that for all your values, the sum of all those differences. Now, you're not really interested whether that's a positive number that you're getting or a negative number, which is why you're doing the squaring and square rooting. If you had a negative number here, you square it and you take the square root of it. That means it just becomes a positive number. Done. Mathematical function to avoid negatives. And then divide this whole thing by how many you had in the sample. We had 3 in this sample. So, oops, wrong. We need to go back to here. So the standard deviation gives you a number for how far away your individual data points are from your mean. That, in a nutshell, is how reliable is your data, not how spot on, how close to the mean are all your data points. Here, that's a huge standard deviation compared to fairly good standard deviations around here, the best one being around 40. Okay, it's not asking you to comment on the data. It's asking you to comment on the reliability of the data, which means you're commenting on this number and these numbers here. So, state one conclusion you can draw from this experiment. One conclusion, only one conclusion. And the one conclusion you can draw is this line here. The mean time taken for 40 degrees is lowest, which means that is your optimum temperature. That is something you can take. Also, the standard deviation is low for this number. Okay. Plus, what you need to clearly say when you're commenting on reliability, that sticks out like a sore thumb, and that is pretty close afterwards. But you definitely need to comment on the temperature, the 10-degree temperature value here, the standard deviation. Those numbers seem to have a wide range between them, and that's just a mathematical number to say how ranged your results are. Right, mark scheme here to go with it. So saying that 40 degrees is your optimum, saying that 10 degrees is a very low reliability data, and a second set of data somewhere. You could as well have commented on the 60 degrees or on the 20 degrees to say that they are relatively unreliable data points as well, considering what standard deviations we've got here. Okay.